Good morning, everyone. My name is John Hierdink, and welcome to the Tribe Public CEO presentation and Q&A webinar event titled our DeltEx Platform, Advancing Innovative Immune Solutions for Cancer and Autoimmune Diseases. It's featuring IN8Bio's CEO, William Ho. As many of you know, I'm the managing member of Tribe Public, and our website is tribepublic.com. That's TRIBEPUBLIC.com. And members from 31 plus countries in all 50 states have joined the Tribe Public freely to gain direct corporate access to leaders and experts that they care about via our webinar events and also via our 36 venue sites located across the U.S., where we host in-person lunch and dinner and speaking engagements with local tribe members. Tribe members furthermore submit names of these experts and leaders of industry that they care about via our free wish list process at our website, adding names of these companies, subjects that span across all sectors so they can learn more with us. Please note that I'm also the managing director of Vista Partners, LLC. It's a registered investment advisor in California, and its website is Vista P Global, VISTAP global.com. Please review both sets of disclaimers at each site and know that I'm a shareholder and an advisor to IN8Bio currently. I would also like to thank all of you for participating for all the questions you've submitted so far, and remind you that you may send in additional questions via the Zoom chat feature during this event, and we'll do our best to get them addressed during this approximate 30-minute event. If we don't get it all done, maybe we'll get lucky enough to have Mr. Ho from IN8Bio back at a further date and/or out to one of the 36 venue sites in the U.S. Like we've done recently. Now let's get started. Will, if you don't mind, if you could start with a little bit of an intro of yourself and what brought you to be the CEO of IN8Bio, and then begin with your presentation shortly after that. Again, thanks for all coming today.
Great. Thanks, John, and thanks, everyone. Very nice to meet you. And for some of the tribe members, very nice to be back in touch again. As John said, my name is William Ho. I am the co-founder and CEO of IN8Bio. Myself, my background, the reality is I never actually intended to be an entrepreneur or CEO. I spent many of my years on the investment side, the finance side on Wall Street, across investment banking, sell-side research, where I was the lead biotech analyst at Bank of America, and on the venture side, where I initiated and ran the public portfolio for a major venture fund, New Leaf Venture Partners, from 2010 to 2014. It was in 2014, which was the summer in which Kite and Juno came public. Kite and Juno were two of the first CAR T companies that ultimately got acquired for combined $21 billion. Those two companies had focused on a technology where we can today take your white blood cells out of one's body, genetically engineer them to specifically target a marker called CD19 to treat B-cell leukemias and lymphomas. Essentially, they were able to create a certain number of cures in their first patients. By the summer of 2014, I was convinced that CD19 targeting CAR Ts were crowded, and I was trying to figure out, how do you ultimately go towards potentially off-the-shelf therapies, and in particular to target solid tumors? It's a market that's 9 times bigger than that of the leukemias and lymphomas, and yet there's been hardly any success to date. It was somewhat fortuitous that I was introduced to our scientific founder, Dr. Lawrence Lamb. Larry, as we call him, was at that point a professor of medicine at the University of Alabama at Birmingham, or UAB. He's a transplant immunologist, an expert in immunotherapy, and an expert in the area of gamma delta T cells. Dr. Lamb has been working with these cells for almost 30 years and is actually credited with the discovery that higher levels of these cells are actually associated with better survival outcomes. And so together we started and launched the company in 2015. Today we are the leading company developing gamma delta T cells in oncology and soon potentially autoimmune disease. In our trials, we're targeting difficult indications, including patients with acute myeloid leukemia or AML and glioblastoma or GBM. And across both phase 1 trials, we have patients who are now over 3 years out who remain in remission despite having difficult target cancers. We believe that gamma delta T cells are powerful immune cells. They've been often called nature's CAR T cell. They have the ability to distinguish between healthy tissue and tumor tissue, killing what's dangerous while leaving what's safe and healthy alone. We believe these cells can help us increase the precision and safety of cellular therapies. And so we are excited about our results to date. We have shown very little additional toxicities on top of the traditional standard of care. We've demonstrated long-term durable remissions, and our team has among the strongest capabilities in this field. Our mission is what we call Cancer Zero, the safe elimination of all cancer cells in these patients' bodies. And so today, as I mentioned, we possess a very comprehensive gamma delta T cell platform. This platform is what we call DeltEx, and this encompasses our know-how, knowledge, and capabilities. These are our trials. INB-100 is what we call DeltEx Allo, or allogeneic or donor-derived gamma delta T cells in leukemia patients. We'll talk shortly about this particular trial and some recent data that we presented at the Tandem meeting, or TTT, in February. Preclinically, we have 3 programs. INB-300 is our non-signaling CAR T program. INB-500 are our iPSC-derived gamma delta T cells. And just last week, we announced our INB-600 platform. It is a T cell engager platform that allows us to directly target gamma delta T cells with a bispecific antibody. And so all 3 of these preclinical programs, we are actively looking for potential partners. And then on the solid tumor side, INB-200 is our lead program currently. It's an investigator-initiated trial in glioblastoma. We presented some data last fall. We'll review that data later today. And we had suspended the phase 2 program, which was known as INB-400, and again, actively looking for potential partners there. To talk about our T cell engagers, this is the newest material that we've announced. We initiated this program somewhat fortuitously last year when we identified that we had a very powerful and potent T cell engager. It's an area that still has a lot of excitement among potential partners and potential investors. Even very recently, there's been numerous T cell engager collaborations that have been completed in the market. Our lead here is to target CD19 as a target, what we call INB-619. CD19 is a widely expressed marker on B cells that can be used to target all B cells. And so we've created an engager. This platform targets on one side gamma delta T cell receptor. On the other side, on what we call an antigen targeting domain, it's a cassette-like structure, so we can swap out the antigen targeting domain listed in green within that green box. And the first target is CD19. We've demonstrated robust depletion of the CD19 target, but what is different vis-a-vis the other gamma delta T cell engagers out there is that historically, gamma delta T cell only encompass about 1 to 5% of the total white blood cell population in your body. We know as tumors progress, the numbers of these cells decline. For the first time, our gamma delta T cell engager can actually drive expansion of that gamma delta T cell compartment. And I'll show you some of that today. And so broadly, a CD19 targeting engager can target both B cell leukemias and lymphomas, such as DLBCL or non-Hodgkin's lymphoma, but also potential autoimmune diseases such as lupus or multiple sclerosis, rheumatoid arthritis, among others. So it really widens the market opportunity for us. And so we developed this in-house. These are the first data that we've presented early last year. On the top These are cytotoxicity data against two different cell lines, NALM6, which positively expresses CD19, MOM13, which does not express CD19. Both of these are leukemic cell lines. We're achieving almost 90% killing in NALM6, as you can see here. These are robust in vitro assays. The E to T ratio is only 1:1, and the co-culture period is only 24 hours. Below, we are incubating with peripheral blood mononuclear cells as a mix, so there are very few gamma delta T cells there. But what you are observing is that with the increased concentration of the engager, we are seeing complete depletion of the B cell compartment. So very exciting again, potent B cell depletion, allowing us to target cells that express CD19 and eliminating them. As I mentioned earlier, we are also driving expansion of gamma delta T cells. Here we have both positive and negative controls with both no treatment and zoledronate, which is a positive control that we know activates and expands V delta 2 gamma delta T cells. But what you're seeing here is that we're achieving up to 450-fold increases in gamma delta T cells. And this expression of gamma delta T cells isn't solely to the V delta 2 compartment, which is quite common on the engager side, but also the delta 1 compartment. The reason why that is advantageous is that V delta 1s are known to be tissue resident, so allowing you to target certain specific tissues, but also a little bit less prone to potential T cell exhaustion and failure. And so we're quite excited about this. Our expansion is about 50-50 between the delta 2 and the delta 1 compartment, and we're getting significant expansion of these cells. This is a very potent T cell engager. This is just showing that we are in the picomolar concentration for the EC50, or the rate in which cytotoxicity you kill half of the cells in the assay. And just visually, here's some results. These are leukemic cells combined with gamma delta T cells, either without engager on the left-hand side or with engager. It doesn't take an expert to clearly see that there are far fewer residual leukemic cells in that image. But even so, many of the residual cells are currently undergoing apoptosis or cell death, with the red arrow showing where the chromatin or the DNA of the cells are disassociated. And so we're excited about this platform. We think our T cell engager has numerous therapeutic advantages, in particular in the rheumatology and autoimmune indications. This is, as I mentioned, this is a pan T cell engager that has demonstrated the ability to specifically eliminate targets in a dose-dependent manner. We can expand both the delta 1 and the delta 2 compartments. CD19 is broadly expressed across all B cells, allowing us to get to deeper responses, potentially driving to an immune reset, which has been observed to be important for potentially curing autoimmune diseases like lupus. As I mentioned, the V delta 1 compartment can resist exhaustion and can target tissue resident B cells for deeper depletion. But ultimately, the gamma delta T cells also provide an advantage over other T cell engaging compounds such as blinatumomab, which is a CD3 targeting gamma T cell engager. Those engagers in the clinic have demonstrated toxicities such as cytokine release syndrome or neurotoxicities such as ICANS, both of which can result in patients being admitted into the ICU and requiring the use of tocilizumab or an IL-6 inhibitor. Gamma delta T cells do not secrete a lot of IL-6. We have not seen neither the cytokine release syndrome nor the neurotoxicities in either of our clinical trials to date. So we have the potential to create a safer, more tolerable therapy in these autoimmune diseases. Finally, we believe in the autoimmune indications. The T cell engagers allow a simpler manufacturing process with lower costs and the repeat dosing that allows us to also avoid lymphodepletion. Unfortunately, the lymphodepletion that requires chemotherapy, for the lymphodepletion that's required for cellular therapy can potentially cause ovarian damage, and many autoimmune patients are women of maternal age between the ages of 20 to 45. So this program has a number of significant advantages, and we will be presenting additional data at medical meetings this spring. But we're quite excited about this, and we're actively also looking for potential partners for these programs. Moving forward in our traditional programs, INB-100, as I mentioned, is a donor-derived or allogeneic program in leukemias. The first observations that gamma delta T cells are associated with better survival outcomes were from our scientific founder, Dr. Larry Lamb. In the 90s, he made the observation that those patients undergoing transplantations that had higher levels of these cells had more than 3.5 times greater survival benefits. And so we sought to make this observation into a therapeutic. Today, we are achieving these higher levels of gamma delta T cells. And the thought was that with transplantation, we can reset the bone marrow in these patients. By resetting the bone marrow, we have a potential pathway towards a cure. However, unfortunately, by about 100 days, 25% of the patients will ultimately relapse. And by 1 year, upwards towards 50% will also relapse. That's because even if I transplant the patient, even if they are what we call minimal residual disease negative, it doesn't mean that there are no residual leukemic cells. All it takes is a single cell to double and to double again, and over time, the patient relapses. Our approach was to infuse these gamma delta T cells so that they can continue to conduct immune surveillance. They continue to float through the blood and they hunt out any residual leukemic cells. And the thought was that if we can do that, if we can continue to keep pressure on any residual tumors, then ultimately the patients won't relapse. This trial was designed as a 3+3 design where we were dose escalating from 1 × 10⁶ towards 1 × 10⁷ cells per kilo. We made the decision to call the second dose cohort our recommended phase 2 dose. We combined AML, CML, MDS, and ALL patients in this trial, and it was run as a single center IIT at the University of Kansas Cancer Center, or what we call KUCC. We are currently in the process of onboarding additional center to get some diversity in the patients. But these are the patients that we've enrolled to date. We presented this data a few weeks ago at the Tandem meeting that was held in Hawaii. We have treated 16 patients to date, many with intermediate or high-risk disease. Many of our first patients should have had a relapse rate of as high as 80 to 90%. We had patients with trisomy of chromosome 8, so an extra copy of chromosome 8, deletion of chromosome 7, or only one copy of chromosome 7, concurrent mutations such as FLT3, IDH, TP53, and others. None of our patients received maintenance therapy. Typically, such patients would receive a FLT3 inhibitor or IDH inhibitor or some other drug. Our patients received a single bolus of gamma delta T cells, and then we observed them. Today, we have multiple patients who remain in remission beyond 3 years. We are demonstrating durable long-term remissions. We have had a few relapses, patient 9 and patient 11. Patient 9 was an ALL patient who went through 7 prior lines of therapy, who ultimately did relapse and pass of their disease. They had a double hit TP53 mutation. Patient 11 also relapsed. That patient was an MDS, MPN overlap patient. They typically relapse by about 6 months. We were able to prolong that time to relapse. And then very recently, we had one MDS patient, patient 15, who also relapsed just shy of 1 year. Importantly, the median follow-up across all of our AML patients to date is 20.1 months, and we have not had any relapses, nor have we had any patients die from progression. We did have one patient, patient 7, who died from complications of their transplant, what's known as idiopathic pulmonary syndrome, patient 7. But how good is this data in actuality? So we went and looked at the retrospective real-world historical control data. We went to the CIBMTR. It is an international center or repository for database of all patients who undergo transplantation This data comes from the United States across all of their AML patients. Across a database of 684 matched patients, CIBMTR demonstrated that at 1 year, 33% of their patients are relapsing and 25% of their patients are dying. We were told, well, the University of Kansas is just really good at transplantation. That's why your numbers are so good. And so we pulled the historical data from KUCC across all leukemias and the AML. I'll focus on AML because AML patients are generally a little sicker, relapse a little faster, and die at a higher rate. When we look at the background of our population, our patients are a little bit sicker. They perform a little bit worse. But again, at the University of Kansas, in their AML patients, they are observing a relapse rate of 43% at 1 year, and 33% of their patients are dying. Whereas, again, we've seen no patients relapse within the first year, and none died to date. The reason why KU's patients do a little bit worse is because they are a major tertiary referral center. They receive complex leukemia cases throughout the Midwest and treat cases that can't be treated elsewhere. So we continue to enroll this trial. We're doing well. We will continue to follow up our patients, and we're on the way towards getting up to 25 patients treated in this trial. And we expect to have additional data updates later this year. Moving forward, INB-200 is our drug-resistant immunotherapy, or DRI program, for newly diagnosed glioblastoma. We presented this data in the fall of last year. To date, we've treated 13 patients, all of them except for one, an IDH wild type. The IDH wild type patients tend to relapse faster. We had one IDH mutant. I will talk about that patient shortly. We had a mix of unmethylated and methylated, 54% of them being unmethylated, meaning that they do not respond to conventional chemotherapy, and a mix of total and subtotal resections. Subtotal meaning the location of the tumor made it unresectable and we had to leave tumor behind. Obviously, those patients typically relapse and progress much faster. The one IDH mutant patient, as of last October, is out beyond 40 months. That's important because last year, for the first time, a drug was approved for IDH mutant gliomas. Vorasidenib is marketed by Servier. They presented data in the fall. In the control arm of their phase 3 studies, the median progression-free survival was 11.1 months, and all patients relapsed by 31 months. And our patient is at over 40 months at this point. But across the board, including both the IDH wild type and the NGMT unmethylated patients, we believe we are extending progression-free survival. This is the data that we presented last fall. When we look across the history of treatment of glioblastoma, unfortunately, 2025 marks the 20th anniversary since the last drug approval for glioblastoma. Temozolomide, or Temodar, has been used since 2005. The median progression-free survival is only 6.9 months, and the median survival only falls between 14 and 16 months after first diagnosis. Our patients received either one dose, marked here in blue, up to 3 doses, marked in purple, or up to 6 doses in the cohort 3, marked in green. Across all patients who received only a single dose, the median progression-free survival was 8.3 months. Every patient relapsed, every patient eventually died. When we look at the patients who received more than 1 dose, so all of those in purple and green, as of last October, the median progression-free survival was sitting at 12.3 months. Already, an 89% improvement over the historical standard of care, and almost a 50% improvement over our patients who received a single dose. And so we do believe we are making a difference. We do believe that with more doses, patients are remaining in remission longer. And we now have multiple patients who have remained in remission longer than what we believe they should have been alive. And so we're excited about this program. We'll present additional data later this summer. But stay tuned. Very exciting data so far. And so to wrap things up, we're coming close to the top of the hour. We're excited about gamma delta T cells. We believe these cells can drive a powerful immune response. We have taken a differentiated approach to cellular therapy. And most importantly, we're making a difference to date. We're demonstrating long-term durable remissions in patients with both AML and glioblastoma, two very difficult and complex diseases, both with patients who are now out beyond 3 years with these diseases. We've been able to extend these time to progression while maintaining the quality of life. We haven't seen any cytokine release syndrome. We haven't seen any neurotoxicities. Any adverse events have been manageable to date, and we're excited. So thank you very much for your time. Thank you for following us, and we welcome you to join us on our mission as we continue on our journey to achieve Cancer Zero. Thank you.
Yeah. Thank you, William. Very, very exciting data. It's amazing the work that you are accomplishing at IN8Bio. Got a few questions and a reminder to the tribe members. You can, if you have any additional questions, just send them through the chat feature quickly, and we'll try to get them addressed. I've got a few of them that you guys have already sent in that we'll go to right now. Just a quick reminder, this is a public company. It trades on the NASDAQ. Symbol is INAB. Indigo Nancy, Albert, Alfred, Boy, INAB. And so you can check that out at your leisure if you're on the investment side here, like many of us are. So again, this is an exciting year. Can you just give us a quick synopsis of what you look to achieve this year from a milestone standpoint? What have you put out to the market already? Could you just briefly go over those points?
Yeah, we expect to have an exciting year. I think in the near term, we're going to continue to provide additional data on our T-cell engager. Again, we said we would present at medical meetings in the spring. We presented some initial data that I showed you today. The first time we demonstrated any of that data was last week at the Cowen Healthcare Conference in Boston. We will show additional data on its ability to kill and expand cells. Sorry, my apologies. Its ability to kill different targets, as well as the ability to kill both oncology targets and potential targets for autoimmune diseases. Mid-year, we expect to present additional data from our INB-200 and potentially the INB-400 program. As a reminder, INB-400 was a phase 2 trial in glioblastoma that was multi-center national. Unfortunately, due to cash resources, we had to suspend further enrollment in that trial last fall. But we did enroll a number of patients, and we continue to treat those patients who have been enrolled and can provide an update there. So we do expect additional clinical data mid-year. And then later in the year, we will expect to have an update on our INB-100 program, which is the DeltEx Allo program in leukemia patients. And we'll spread some additional presentations at different investor meetings and medical meetings and elsewhere throughout the fall as well.
Thank you, William. Speaking of banks around the company, you've completed some successful financings over the last year. In addition to that, do you have analyst coverage? What banks are covering you at this point?
We do have a number of banks who cover us. AC Wainwright covers us. Rodman & Renshaw covers us. Mizuho covers us. Jones covers us right now, as well as Laidlaw.
Okay, well, that's pretty good.
Yes, and there's Zacks also covers us. Sorry, there's 6 analysts right now.
Okay, gotcha. And then are those buy ratings, or where are they?
They're all buy ratings, and I believe the median price target is probably somewhere around $8 right now.
Okay, and if over the next year to 2 years, as you're moving through this window, you've mentioned sort of partnering possibilities. And I know no one really knows exactly when that timeline can happen, because it takes 2 or 3 to tango, depending on the situation. But if you, in a heart of heart, how do you feel about the possibility of doing that based on your extensive background and in the space and the deals? Is this from the data standpoint, is there at one point that really thinks that triggers that process to some degree, or what would you, any color you could?
I think it's a continuous dialogue. We have continued to have dialogue with potential partners, both large and mid-size, across our pipeline. Some of that over time waxes and wanes depending on the market environment, depending on their own budgets, and depending, unfortunately, sometimes based on the success or failures of our competitors. As everybody knows, it has been a tremendously difficult time for allogeneic cell therapies in the market over the last couple of years. We have numerous competitors who have failed to demonstrate durable remissions. We've had some competitors who've had toxicities that have resulted in, unfortunately, patient deaths. But we as a company have continued to execute. Maybe it's a little boring. We come back time and time again so far and have given updates on our patients, and many of them continue to be in remission and continue to do well. We've had multiple patients, we've had multiple patients in some cases go back to their occupations, go back to work, and in some cases they're currently working when, in our belief, they should have been deceased already. And so we're going to continue to execute. It's been a difficult, not only micro environment in biotech, but also, as you know, macro environments. And we're hopeful, we're actually optimistic that as we get through this year, we'll go through this painful period right now and we'll see light on the back half.
Okay, thank you. As far as, you haven't, I guess, filed at this point, so you may not be able to answer this, but as far as the cash reserves and sort of projections for this year, have you put out, what is the latest you put out there? And can you speak to, where you can take the company, and if anything's changed here?
Thanks, John. So you are correct. We haven't announced the fourth quarter earnings yet. That will come out shortly as it's required. I think the last guidance that we gave was that based on the cash that we had on hand, we can get through 2025. Okay. And we have a lot of milestones still, lots of opportunity, and we're going to continue to execute. And we'll have those numbers and some guidance out shortly.
Yeah, we've seen the company truly, at least its stock side of it, has taken off recently. You had a massive day recently where it traded over 224 million or something in that area, and the stock just flew up. I think it was 90% at one point, and that was based. It moved on some exciting data that you put out. So yeah, I could, and you've got relatively great liquidity for a small company. So it'd be interesting to see as you roll out these milestones this spring, which we're in it. I guess it's rather imminent, being it's into March here. That could be an exciting spring for us. So, and both on the stock side and fundamentally in the company, as you reveal at these conferences.
Look, we will be in conferences and out of conferences and reveal some new data. We're excited. Look, ultimately, despite what happens on the macro environment, our job is to execute and deliver data. We do believe that long term, if we can keep patients alive, ultimately we will be rewarded. That's what matters in the end.
Well, since it's at the past the top of the hour, I wanted to again thank you for joining us today, and thank all the tribe members that came along and spent their little bit of their morning or afternoon, depending on where you are in the world, with us. And I want to remind you that, again, part of our process in learning is, in these tribe events are across these 36 venues here in the U.S. And so if you do have interest in meeting William and learning more about IN8Bio, again, the NASDAQ symbol is INAB, express that through the wish list process at the website, or ping me if you have my contact information at John, John, at tribepublic.com. And I'll do my best to see if we can get William somewhere close to you and an event coming up soon. And so continue to express your interest there. And if there's something we didn't get answered today, send us that. And if you're not in one of those 36 venue sites, let us know. Maybe if you're in the US, we can try to create a tribe around you. And again, if you are outside the US and you want to learn more about IN8Bio, ping us there on the wish list too, and let us know that you want him back on a webinar sometime in the near future as he reveals some of these milestones and fundamental advances in the company, and we'll continue to learn. William, I want to thank you again for joining us today and IN8Bio, and giving us some more insights as to why you're so excited about the company, and further justifying the investment I've made. And we look forward to working with you in the future. And thanks again, Tribe, and I'll bid you guys adieu, and we'll look to see you on. Again, one last reminder is I'll look to get this event video up at the Tribe Public YouTube channel later today. And you can, again, watch some of that, and please share it with other members around the world, and those that may be not aware of the Tribe yet, and IN8Bio. So again, thank you all, and I look forward to seeing you again soon on the next event. And thank you again.
Great. See ya. Thank you.